Hello students, welcome to EPG Pathshala. I am Dr. Bihu Prasad Panda from School of Pharmaceutical Education and Research, Jamia Hamdard, New Delhi. Today we are going to discuss about a module regarding microbiological assay of antibiotics under paper Pharmaceutical and Biological Analysis. In this module, we are going to learn about microbiological culture medium, test microorganism for antibiotic assay, apparatus used for antibiotic assay, the assay design, the cylinder plate or cup plate method, and the turbidimetric or tube assay method. The inhibition of microbial growth under standardized condition may be utilized for demonstrating the therapeutic efficacy of antibiotics. Any subtle change in antibiotic molecules, that is in chemical structure, which is not detected by chemical method, can be revealed by a reduction in the antimicrobial activity. The microbiological assay is based on comparison of antimicrobial activity. The microbiological assay is based upon a comparison of inhibition of growth of microorganism by measured concentration of antibiotics to be examined with that of produced by unknown concentration of a standard preparation of the antibiotic having known activity. Two general methods are usually employed for antibiotic assay. Number one, the cylinder plate or sometimes called cup plate method is also known as method A, and second method is the turbidimetric or also known as a tube assay method, that is also called method B. Preparation of microbiological media. How we can prepare the microbiological media to carry out antibiotic assay. The microbiological media required for preparation of test organisms are made from the culture media ingredient like beef extract, yeast extract, peptone, sodium chloride, agar, etc. The different composition of media is given in table number 1. Minor modification of the individual ingredient may be made or reconstituted dehydrated media may be used, provided the resulting media have equal or better growth promoting properties and give a similar standard curve response. Dissolve the ingredient in sufficient water to produce 1 liter and add sufficient 1 molar sodium hydroxide or 1 molar hydrochloric acid as required, so that after sterilization the pH of the media will remain in between 6.5 to 8.0. The liquid media or solid media were prepared in culture tube or in petri dish as per the assay design. Composition of microbiological media used in antibiotic assay. You can see here the media is made up of different ingredients, and these media components are different for different media type. For example, in medium A, that is medium A microbiological assay medium, you can see the ingredient here, that is peptone, pancreatic digest of casein, yeast extract, beef extract, dextrose and agar, and the pH of the media is maintained at 6.5 to 6.6 after sterilization, whereas you can see the media or the microbiological medium B, the composition of medium B is different from medium A, and similarly the medium C having a different composition, different ingredients, whereas medium D having some other components, medium E having other components. It depends upon the what type of antibiotic, what type of procedures we follow in assay, or it is mentioned The media components are used as per the pharmacopoeial rule. The pH of the media is very important. The pH of the media after sterilization must be in between 6.5 to 7.3, or in some case it is 8, for example in medium E, and the pH of the media is kept as per the pharmacopoeial rule. Agar is used when we need a solid media, whereas for liquid media, for example in medium C, we do not use agar because agar is a solidifying agent, and the liquid media is most commonly used for method B, that is a turbidimetric method, whereas the agar containing media are most commonly used for cylinder plate method. Preparation of standard solutions, that is standard antibiotic solutions, and unit of activity. To prepare stock solutions of antibiotic, dissolve a quantity of standard preparations of a given antibiotic accurately weighed and previously dried in the solvent specified, and then dilute to the required concentration as indicated as per IP. Store in a refrigerator and use within the period indicated as per IP. The potency of the standard preparation is explained in table number 2, is expressed in international unit or microgram per milligram of the pure antibiotics. The standard preparation for India are maintained in Central Drug Laboratory, Calcutta. Standard solutions of antibiotic and unit of activity. You can see here in the table number 2 that the antibiotic name is given and standard preparations are given and its activity and mg of antibiotic containing 1 unit of antibiotic activity. For example, bacitracin, the standard preparation is zinc bacitracin having activity 74 units per microgram per milligram, and 1 unit of bacitracin is equal to 0.01351 milligram. Test microorganism and inoculums. What type of test microorganism we can use for antibiotic assay, and how the inoculum or the seed culture of the microbe are prepared? The test microorganisms are different for different antibiotics, that is explained in table number 3 and 4. The microbial suspension were prepared as per the method given in table number 5. For cylinder plate or cup plate method, that is method A, selection of test microbe. The inoculum is inoculated in media plate. The cylinder is filled with medium concentration of antibiotic solution and incubated. After incubation, measure the zone of inhibition, the volume of suspension that produce optimum zone, that is clear, and the diameter of the zone is determine the inoculum to be used. For turbidimetric method or for method B, selection of test microbe is carried out. Microbe or the inoculum is selected based on the optical density obtained between low and high antibiotic concentration. Test microorganisms. Test microorganisms are specific for specific antibiotic assay. You can see the table number 3 and 4 that shows the different antibiotics and microorganisms, specific microorganisms. These microorganisms are obtained from different culture banks, specified culture banks as per the pharmacopoeia. Most commonly, the microbes are obtained from American type culture collection, or it is called ATCC. Microbial suspension. The microbial suspension used for antibiotic assay are specifically mentioned in pharmacopoeia. As per the pharmacopoeia, we have to prepare the microbial suspension for specific antibiotic assay, and this is given in table number 5. To carry out microbiological assay or antibiotic assay, we need to use certain apparatus or instruments. All the equipment or apparatus used for antibiotic assay must be cleaned before and after use, because this is necessary to maintain the sterility and reproducibility of the result. Glassware for holding and transferring test microbes must be sterilized by dry heat or by steam so that we can carry out aseptic work. Different apparatus used for microbiological assay are incubator. Incubators are mainly having temperature control system. In some incubator, you may find also humidity control systems, but for microbiological assay or for antibiotic assay, we use a temperature control incubator systems. These incubators are used for culturing microbes, for preparation of inoculum, for preparation of seed culture, incubation of plates or culture tubes. Spectrophotometer, this is another apparatus, are used for measurement of microbial growth at a different wavelength, particularly 600 to 620 nanometer of wavelength is used for measurement of the bacterial growth. But as per IP, we can use as specified wavelength. Another apparatus, that is cylinder is used in method A or in cylinder plate method of antibiotic assay. Cylinders are used for holding antibiotic solutions. Antibiotic solutions may be samples or may be standard. The cylinders are having 8 mm outer diameter, 6 mm inner diameter and length of 10 mm. The cylinders are made up of glass or steel, porcelain or aluminum, but most commonly glass and steel cylinders are used in antibiotic assay. Other apparatus or instruments includes autoclave. Autoclave is apparatus which is used for preparation of sterile medium. Hot air oven is another apparatus used for sterilization of glasswares by dry heat method. Laminar airflow unit. Laminar airflow units are used for aseptic work. Petri dish, culture tubes are the glasswares are commonly used in microbiological assay system. Cylinder plate or cup plate method of antibiotic assay. This is also known as method A. This method depends on the diffusion of an antibiotic from a vertical cylinder or a cavity through the solidified agar layer of a petri dish. The growth of added microbe is prevented entirely in a circular area or zone around the cylinder or cavity containing a solution of antibiotic. It may be standard or it may be sample. Procedures or experimental procedures to carry out cylinder plate antibiotic assay. Step 1. Inoculate the sterile liquid media with microorganisms, that is a test microorganisms specified as per pharmacopoeia at a media temperature between 45 degree centigrade to 50 degree centigrade so that the microorganism will remain live. This is very important to have a proper temperature while adding the microorganisms into sterile liquid media. If liquid media is having higher temperature above 50 degrees centigrade, the microorganisms may not survive, whereas at lower temperature below 45 degrees centigrade, the liquid media, that is molten media, may solidify due to gelling effect of agar. Step number 2. After inoculating the sterile media with microorganisms, immediately pour the inoculated media into petri dish with depth of 3 to 4 mm. The thickness of the media must be uniform. Otherwise, if there is a uneven thickness, the result will be incorrect. There is need to pour the medium into petri dish. Since the media is under molten condition, if it is delayed, the media start to solidify and it will form a uneven thickness of layer. Step 3. Prepare the antibiotic solutions using appropriate buffer solutions as per the pharmacopoeial procedures. Step 4. Apply the antibiotic solutions and test solutions over the agar surface using cylinder. The volume of the antibiotic solutions or test solutions must be uniform. Step 5. Keep the petri dish at 4 degrees centigrade for 1 to 4 hours for diffusion of antibiotic suspension into agar medium. This is very important because the antibiotic suspension, that is standard or samples, must be diffused into the agar medium. Otherwise, the zone of inhibitions cannot be obtained. Step number 6. Incubate the petri dish at 32 to 37 degrees centigrade for 18 hours in an incubator. The temperature of the incubator is selected as per the pharmacopoeial procedures. Step 7. After the incubation is over, measure the zone of inhibitions and calculate the result. Results can be calculated by using the formula L is equal to 3A plus 2B plus C minus E divided by 5. H is equal to 3E plus 2D plus C minus A divided by 5, where L is the calculated zone diameter for lowest concentration of the standard curve response line. H is the calculated zone diameter for highest concentration of the standard curve response line, whereas C is average zone diameter of 36 reading of the reference point standard solutions. A, B, D, E are corrected average values for the other standard solutions lowest to highest concentration respectively. Turbidimetric or tube assay method. This method is also called method B as per Indian Pharmacopoeia. This method has the advantage of a shorter incubation period for growth of test microorganisms. It takes usually 3 to 4 hours, but the presence of solvent residues or other inhibitory substances affect the assay result more than the cylinder plate assay. For turbidimetric or tube assay methods, we use spectrophotometer to measure the bacterial growth. Methodology for turbidimetric assay. Step 1. For turbidimetric assay method, prepare 5 different concentrations of the standard solutions. For preparing standard curve by diluting the stock solutions of standard preparations of antibiotic as per the pharmacopoeia. After preparation of the standard solutions, select the median concentrations and dilute the solutions of substance being examined, that is unknown substance, to obtain approximately this concentration. Step 3. Place 1 ml of each concentration of the standard solutions and of the sample solutions in each of the test tube in duplicate. Step 4. To each tube, add 9 ml of nutrient medium previously seeded with the appropriate test microorganisms as per the pharmacopoeia. Step 5. At the same time, prepare 3 control tubes, one containing the inoculated culture media, that is culture control, another identical with it but treated immediately with 0.5 ml of diluted formaldehyde solution that act as a blank, and a third containing uninoculated culture media. Step 6. Place all the tube or culture tube randomly distributed or in a randomized block arrangement in an incubator or water bath, and maintain them at a specified temperature between 32 to 37 degrees centigrade for 3 to 4 hours as specified by pharmacopoeia. After incubation, add 0.5 ml of dilute formaldehyde solution to each tube to stop the growth of the bacteria or the microbes. Step 7. Measure the growth of the test microorganisms by determining the absorbance at about 530 nanometer of each of the solution in the tube against the blank. Estimation of potency after the turbidimetric assay. After the turbidimetric assay, the estimation of potency is carried out by using formula L is equal to 3A plus 2B plus C minus E divided by 5. H is equal to 3E plus 2D plus C minus A divided by 5, where L is the calculated absorbance for the lowest concentration of the standard response line. H is equal to the calculated absorbance for the highest concentration of the standard response line, whereas A, C, B, D, E are average absorbance value of each concentration of the standard response line, lowest to highest respectively. Before antibiotic assay, before carrying out any antibiotic assay, we should learn how to prepare the media, how to handle the laminar flow unit, how we can subculture the test microorganisms, how we can transfer the test microbes into media in a sterile manner, how we can handle the contaminated medium, and how we can dispose the culture medium after the experiments are over. Without knowing these skill operations, we cannot able to carry out a validated antibiotic assay. Preparation of sterile microbiological medium. The steps involve accurate measurement of medium component. For solid medium, solidifying agents, that is agar, is added, whereas for liquid medium, agar is not added. The solid culture medium were prepared in the form of slants or in petri dish, whereas liquid medium were prepared in culture tube for suspension culture. The medium component include agar were dissolved. The medium component including agar were dissolved by heating in a water bath or by using a microwave oven. Next, the pH of the media is adjusted to required pH by using 1 normal NaOH or 1 normal HCl, 1 normal hydrochloric acid using pH meter or by pH paper. The glassware used were plugged with non-absorbable cotton and wrapped with aluminum foil before sterilization. The glassware containing culture medium were sterilized by autoclave for 15 minutes at 15 psi pressure. Working in a laminar flow unit. It is very important to know how to work in a laminar flow unit to carry out any aseptic work. Laminar flow units are used for any aseptic or sterile work. The environment inside laminar flow unit is made sterile by using ultraviolet lamps, using spirit lamps, sterile air and ethanol, that is 70% ethanol. Before using laminar flow, the surface of the laminar flow unit is cleaned by ethanol, that is 70% ethanol. The material, that is culture medium, glassware, were kept inside the unit except the live microbial cultures. Then the UV lamp or the germicidal lamp is kept on for 15 minutes. After 15 minutes, the UV lamp is switched off and white light is on, and simultaneously the airflow is also on. The surface of the unit or laminar flow unit is again cleaned by using ethanol, that is 70% ethanol. After drying of ethanol from the surface, the spirit lamp is made on inside the laminar flow. Next, the hands of the worker is cleaned by ethanol. Now, we can start the aseptic work in laminar flow unit. Subculturing and storage of test microorganisms. Here, this is the diagram showing how we can subculture the microbe from one culture media to another culture media. That means from used cultures media to a fresh culture media. You can see here a loop is there, two culture tubes are there, one is having microbial growth and another is blank, and one lamp, that may be a spirit lamp. Now in step 1 you can hold the culture tube in one hand and in other hand you can hold the inoculating loops. In the step 2 you can see we can sterilize the inoculating loop by using spirit lamps to red hot so that the inoculating loops will be sterile. In the step 3 we can open the mouth, we can open the holder or plugs of the culture tubes in front of flame so that there will be sterile environment inside the tube as well as outside the tube, and we can carry out the aseptic work. Next step, we can carry out the aseptic work within 15 cm radius of the flame. First, we can dip the inoculating loop inside the culture tube in which already the microbes were grown by. In the next step, the same inoculating loops can be transfer the bacteria into fresh media. After transferring the bacteria from one culture tube to other culture tube, in presence of flame, we can close the mouth of the tubes or we can plug it by using non-absorbent sterile cottons, and after that we can again sterilize the inoculating loop. This is how we can carry out subculturing of microorganisms from one tube to other tube. Storage of microorganisms. This is very important to know how we can store the test microbes. The test microorganisms can be stored by any of the following method. Number 1, by using slants at 4 degrees centigrade for 1 month. That means The microbe can be stored for 1 month at 4 degrees centigrade in agar slant or in nutrient slants. Microbes or test microbes, particularly spore-forming microbes, can be stored in sterile soils for 6 months. The test microbes also can be freeze-dried or lyophilized and can be stored for many years, for 5 years, can be stored for 5 years. Sometimes we also store the slants containing microbe by overlaying with mineral oil. Sometimes we also store the test microorganisms by cryopreservation. We can also store the microbes by cryopreservation, means in a liquid nitrogen container. Sometimes we also store the microbe in 20% glycerol stock at 80 degree centigrade, at minus 80 degree centigrade. Transferring of a test microbe into medium. Now, how we can transfer the test microbe into medium? This is very important because when we carry out antibiotic assay by using cylinder plate methods, we follow this method, that is transferring of test microbe into medium. First of all, the molten agar medium is prepared, and to this the original microbial culture or test microorganisms are added at a temperature between 45 to 50 degrees centigrade, and this mixture is very well mixed, and then after that it is poured into empty petri dish in a laminar flow unit. This method is also known as pour plate method. After solidifications of media, the media along with microbes can be used for antibiotic assay. Handling of a contaminated medium and disposal. It is very important to know how we can handle the contaminated medium and how to dispose the medium after the experiment is over. The biological waste include but not limited to petri dish, pipettes, culture flask, syringe, needles and culture media, whatever we used in the experiments. All biological waste, including non-infectors and infectors generated during laboratory research, must be deactivated by autoclaving or by chemically treatment before disposal. It is suggested that decontamination should be carried out by autoclaving at 121 degrees centigrade for 60 minutes. After autoclaving, media may be disposed of through normal waste disposal or by incineration procedure. When disposing of slant cultures, remove the cap and turn the tubes upside down in the baskets. Place the cap in a separate container, then separate the slants tube by size. So students, let us summarize what we have learned from this module. In order to get correct and validated results, the microbiological antibiotic assay results must be validated by statistical analysis. Microbiological antibiotic assay must be carried out as per the pharmacopoeial method. The standard antibiotic which is used in experiments must be procured from authentic source. In India, we can procure it from CDL, Calcutta, or as directed by Pharmacopoeia. The microorganisms or the cultures must be obtained from ATCC, that is American Type Culture Collection, or from NCTC or from NCIB as per the pharmacopoeial directions. The culture medium must be from authentic chemical source. Correct SOP must be followed for handling the culture, for aseptic work and post-experimental work. Fiducial limit. The fiducial limit of error of the estimated potency should be not less than 95% and not more than 105% of estimated potency. The microbiological assay has been used extensively to estimate the potency of growth inhibiting substances by comparing their quantitative effect with a reference standard of defined potency to establish pharmaceutical equivalence of new generics, new molecules, and as manufacturer quality control. The actual inhibition of the observed microbial growth under stringent standardized experimental parameters may be judicially utilized and adequately exploited for demonstrating as well as establishing the therapeutic efficacy of antibiotics. Thank you.